This is TWIS. This Week in Science, episode number 1071, recorded on Wednesday, August 12th, 2026. 5 slimes to rule them all. And 1 slime to bind them? I don't really know. I'm Dr. Kiki, and tonight we will fill your head with red spots, snail slime, and primate brains. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. Fauci in contempt of Congress, the National Academy's capitulating on a climate chapter, more vaccines are optional while MMR might be split into 3, and unproven peptides are more available than ever, maybe? Political pet projects have taken over the hill regardless of scientific support. At least we still look to the evidence here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And good science to you, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back again to talk about all the science that was fit to spit in this episode of this podcast. I mean, it's not really fit to print because we don't go to print. So, yeah, I don't know. I got to work on some of these words sometimes.
No, it sounds to me like the plan is to say it and spray it, and that's fine because none of you are actually physically here with us.
I know, and I have a pop filter, so hopefully that helps
There
a little
you go.
bit.
Yeah.
I hope everyone had a wonderful week. We're back, ready to talk about all the science that we brought tonight. I have stories about some unnatural viruses, black hole star. Yeah, that's as far as I'm going to go with that one. Bacteria for babies, maybe some Elven coral, some monkey brains, some trust tests, and the LLM study that we really all needed but didn't know we needed.
Excellent.
Yeah.
Excited for that.
What's in the animal corner, Blair?
Platypus, frogs, snails.
It sounds like an animal corner.
Yeah.
Yeah, sounds like it's rounded out as well. You've got some verts, inverts,
Yeah.
and
Monotreme. Yeah.
Monotremes, amphibians. We get so many good groups
It's
here.
literally 3 of my favorite animals also. This is pretty
Oh.
exciting.
So Blair's going to be waiting impatiently for her turn for her animal corner.
I'll be patient. It's fine.
I see how it goes. All right. As we jump into the show tonight, I do want to remind everyone that if you have not yet subscribed to This Week in Science, you can find us live streaming on our channels weekly Wednesdays, 8 p.m. Pacific time-ish. On Wednesdays at 8 p.m. I said that. Wednesdays, 8 p.m. Pacific time-ish, right? On YouTube, Facebook, and Twitch. I started hearing doors closing and things happening in my house, and I got off kilter there with what I was saying. Anyway, you can also find us on your favorite podcast platform. You can look for This Week in Science, look for TWIS, and you will probably find our wonderful orange starburst flag and be able to subscribe so that you can find us every time a new episode is published. If you want information about show notes, the stories we talk about, please head over to twis.org, our website, where we also have links to our Patreon account and our Zazzle store. But now it is time for the science. Should we jump in? Let's do it. Okay. Story published in Science this last week, and a bunch of articles came out about it. Carl Zimmer wrote about this in the New York Times, and there's a great article if you're not able to get Carl Zimmer's wonderful guest link through the New York Times. There is another great article by some scientists in The Conversation. Now, what is this story? Researchers published in Science their work titled Generative Design of Bacteriophages with Genome Language Models. What does this mean? Scientists have used large language model technology trained on genomic information about bacteriophages, very specific genetic data sets, and use that to create synthetic bacteriophages. We'll just shorten that and say phages from here on out. Now, it's been simplified and said it's a virus. Well, that's because there's like everything that's small and infects things and is not really living, quote unquote, it's a virus. It's not bacterial. These bacteriophages, they're viruses. They are, or bacteria, actually, I'm sorry about that. They are bacteria viruses. Bacteriophages are little tiny viruses that infect bacteria.
Mm-hmm.
And they're really cool because we didn't know about them for a long time. And then we're like, oh my gosh, this is where the predator-prey arms race for antibiotic resistance, all sorts of things. Like we have found our antibiotic targets very often because of how they are similar, very important targets that are targeted by these phages very often. And there are even uses in what's called phage therapy for really hard to kill infections, where they're using phages, where they've found very specific phages that infect very specific types of bacteria at very specific targets, and they're able to use multiple phages to be able to get rid of multi-drug resistant bacteria. And so this is really important as we're moving into a future without enough really well-working antibiotics. Okay. So first thing about this story, very interesting use of what we call a language model, right? They have taken that model system, the training, it's not language, it's genetic code, right? So they've basically done a similar kind of training set. Just the language is base pairs and genetic data. And so they didn't just say, here's all the genetic information in the world, make up a new bacteriophage. They did not do that. They were very, very specific in their target. What they did is they honed in on one very, very specific natural phage that has a specificity for E. coli. This phage is Greek letter 174. I don't know how to say this. Omnicom X174. Anyway, this phage is a template and was the design template for their genome language models that are called EVO1 and EVO2. They used all these EVO1 and EVO2 models were trained on a bunch of phage genomic data for genetic architecture, specificity to host. So basically this type of structure of information goes with E. coli. This goes with Salmonella. This goes with this. And so they created, using X174 as a template, they basically created a way to pump out new designs, basically to speed up the process of evolution. So instead of relying on the natural process of things just bumping into each other and working to infect bacteria and to reproduce themselves using bacterial mechanisms, they don't have to do that anymore. They can potentially make these viral bacteria viruses in a laboratory. They tested nearly 300 chemically synthesized phage genomes. They got 16 that worked. And those 16 were very specific to the host. They had different fitness levels, and they had competitive infection kinetics. And so that's important because that determines in an ecological system, a microbial bacterial ecological system, the movement of how different phages are going to be able to infect or not infect in a certain environment. Anyway, they were able to do this. They had generated phages also overcome bacterial resistance. So if the bacteria became resistant to phage antimicrobial therapies, they gave them a mixture of different designed phages, and the mixture was able to end up with survivors and to be able to outcompete bacterial resistance. And so this is really important for that predator-prey interaction that goes on. So really interesting, great story, lots of cool stuff that is going on with this. The concern is this was very easily managed by open source genome language models, and there really aren't any conversations going right now about who regulates and how this kind of technology is regulated if it does become regulated. Where, when, and how is this technology allowed to be used? Because not only can you use it for therapies and attacking bad bacteria, you could also
make
destroy
a superbug. Yeah.
You could boost up a bacteria. You could destroy good bacteria.
Yeah.
You could do so many different things. And so there is obviously a concern that comes along with it.
I looked. It's the first Greek letter is the word phi for phi.
Thank you.
And I don't know if it's X174 or Chi 174.
That's what I was like.
Yeah.
I don't know. I don't know for sure, but
hey, there you go. Okay, so this is all still very proof of concept. It sounds like
it is very proof of concept.
Yeah.
There are so many reasons this is still far away from actually being used in medicine, right? Or in, beyond a laboratory at this point in time. This is still, years in development. You have to make sure you can test these things. How do we even develop a process for ensuring that these kinds of phage therapies, synthetic phage therapies, could be repeatable and, nimble enough and safe enough to use nimbly in a hospital setting or in an off the shelf? How do you make it off the shelf? How do you make it something that works really well? But I think it's a really exciting development in the use of AI for science, for medicine, for speeding up the possibility of finding therapies and uses, that, otherwise we have to search through all the swamp soup.
Yeah. Yep. Absolutely. I mean, that's what LLMs are good at, is processing data and creating iterations. Like, that's what they're good at, because those are the things that we would have to sit and do manually that an LLM can really handle. If you give them a very clear set of parameters, they got it.
And of course, they said they came up with lots of different options for genetic different genomes for phages. 316 of them worked. So it's not like, it's just going to design a great one every time. There's still testing and iteration and all sorts of stuff that needs to be involved. And the article on the Conversations website does a really good job of going into the gap that is still existent between designing a phage and treating a patient. So, but the question is, for biosecurity, how come we don't know about people already having this conversation, right? Why don't we already have regulations about this? We should. Anyway. Yeah. I think this is my cool news that's going on that everybody should be aware of, because this might be where bacterial infections are treated or how they're treated in the future. you know, how do we get there faster? But there are safety issues, so it's promising. Yeah. What about platypuses?
Platypus. So, platypus, Australia, monotreme, egg-laying mammal, crazy guys. There was a drought in 2019 in Tidbinbilla Natural Reserve in Canberra. Of course, Tidbinbilla sounds so Australian.
Tidbinbilla.
Yes, there were. They only found 8 platypuses who had passed, and then they found an additional 7 who were still alive. They rescued the 7. 5 survived long enough to then be returned to the wild. But they didn't know whether spending literally months in captivity after having been born in the wild would affect their ability to reestablish. That is often the case, especially with mammals. Once you bring them in to live with humans, especially kind of in a slapdash approach where it's, you know, you gotta kind of just act fast. You're not thinking about like, oh, let's get a platypus puppet and put them in a dark case. And then like, you know, like they were just like, animals are dying, we gotta grab them. So they grabbed them. There's a chance that they'll habituate to humans. They won't want to get food on their own. They'll keep approaching humans, or they just plain won't survive in the wild afterwards.
We hope the last one is not the case.
Right. And so this was a really unique opportunity where they were able to then follow the platypuses post re-release. They followed them for 18 months after they were returned to the nature reserve. They were at the Taronga Zoo for 5 months in human care. They found that the animals quickly reestablished their homes. They resumed normal movement patterns. They settled back into familiar parts of the waterways. All within days of their release, they started acting pretty normal. 4 of the 5 animals remained in the reserve for 10 to 18 months after the release, so it seems as though they were reestablished successfully. This is just good evidence that the emergency rescue and return can be an effective conservation tool for platypuses. You know, there's, like I said, lots of different issues with doing this with other species, but there's not a lot of research on platypus. So in this case, looks like the platypus is an animal that you can rescue, treat, bring back to health, and then release back into the wild. They settled back into their regular routine, and they served their evolution or ecological purpose in that space. So it seems like they got right back into it. So this just will kind of provide one in the win column for this. It encourages researchers to try to do this more, conservationists to do this more with other species, and that at least in theory, based on this very small sample size, platypuses you can do this with, and you can boost their survivability if there's a natural disaster, which obviously there's more of due to climate change and stuff like that. So
yeah. So I'm like, they've had conservation efforts for various birds, various mammals, various, you know, monotremes are this. Did they treat them? How did they treat them?
It was a drought. So I'm gonna guess.
Like with birds, you feed them with sock puppets, and you try to get rid of all your human smell. It sounds
like they just brought them into the zoo. So they just fed them and gave them water and shelter and allowed them to cool off and gain body weight back. Then they released them back into the wild. Yeah. So they basically just sheltered the drought because, you know, they're aquatic animals. So in a drought, if there's no place for them to go, there's no food for them to eat, that's it for them. So
hopefully they're able to do this, you know, much more. And I mean, why is it Australia, New Zealand, these island continent countries, you know, there's so many. Is it because they're isolated that they have? Do they have more animals like the platypus and others that are needing this kind of conservation? Do they have more endangered animals?
So, I mean, generally speaking, if you're on an island, there's less space to move to adapt to a rapidly changing climate. So island animals in general are more susceptible, just like mountain animals are more susceptible.
Yeah, okay.
That's like a geographical island, right? Because you can't go down to exit out into larger territories. Your mountain is all you have. So in a mountain, in an island, you are really constrained geographically. And so, you know, if I was a polar bear and I was hanging out in the lower parts of Canada, I can scooch further towards the North Pole as the climate warms. But if I am a platypus in Australia and I need fresh water, and there's no fresh water in direct relation to where I am, there's nowhere else I can go, right? So generally islands are more difficult for that, but I think also Australia's really unusual in that they have these like very intensely varied ecosystems. They have freshwater ecosystems, they have deserts, they have mountains, they have beaches, they have like everything in Australia in this pretty geographically small space. So if you have an impact on one kind of like ecological biome, they can't go next door. They can't go to another neighboring area because it is ecologically so different in that other space. So I think it's just a function of like the biogeography of Australia. But also the last thing I'll mention is that because there's so many weirdos in Australia, like there's all these evolutionarily weird animals. You know, some people would call them primitive, but just basically that certain traits have maintained that have been selected out on the mainland. And because of that, that also provides less plasticity in all of their behaviors and needs, because they don't have as varied of a genome with like all this evolutionary history of change. A lot of things have been maintained for millions of years. And so there's way less opportunity for them to adapt to the new space.
Right.
Because they haven't had as much ecological like pressure to evolve as mainland animals have. So in general, like Australia is special. We know that.
We know that. Yeah.
And their animals are special and they're unlike anything anywhere else in the world. So also that's part of it. You can't be like, oh, well, if we lose this population of platypus, we'll just grab some from South America and bring them over. No. This is
different platypus. Yeah.
This is all of them. So like you got to save them. You got to save these habitats and these populations. And if they can be taken in and taken care of and re-released, golden. That's great.
I mean, this makes sense. The emergency rescue makes sense to me, especially of adult animals, because adult animals have already imprinted on their own species as mates. They have already learned their wild habits, feeding habits, habits for predators and vigilance, and you know, all they've learned their languages, right? The language of their species and the other animals in their environment. So there's a part of me that's like, I'm not really surprised that they did okay.
There's certain animals out there that if you like, bears are famous for that. Like if you brought a bear inside and you taught them that they could take food from humans, toast.
All done.
Yeah.
Nope, they're always going to
hunt and gather. Absolutely not. Are you kidding me?
Do it anymore.
I could just go over there and take food from that guy. No.
I just saw and this made me think. And of course, it's like social media videos. People can edit any which way, right? But there was a video that somebody had edited together various clips of a crow hanging out with a guy, and I don't know. I'm sure that if I showed it to my son, he'd be like, that's AI. But but the whole thing was like, this is so sweet, you know, hearts and tears. This guy noticed a crow who was thirsty out in the environment and gave him water. And so the crow followed him home. And then the crow hung out with him. And so then the guy fed him. And then the crow never left. And now the crow jumped on his shoulder. And now he rides with him on a bicycle and they're best friends, right? And I was like, I mean, I can imagine a crow giving up independence for somebody who just fed it. You know, the crow's smart enough to do that. Take advantage. You know, but at the same time, I was like, this just it gives that like the wrong-headedness, right? The wrong visual about how humans should interact with wild animals. And so, especially with a rescue story like this, I think it's very important to say, don't feed the animals unless
No.
you are a trained professional who is in a situation of helping them on purpose.
Yes. I mean, listen, we would all love to be Dr. Dolittle or Snow White or whatever fantasy character you want to imagine that's with the animals. But
right,
for every animal that is taken out of the wild, that is an entire life history of that animal that you are taking out of the wild. Even if you could give a perfect life to that animal, which you probably can't.
You can't.
Right? But even if you could, their children, their children's children, their children's children, every plant they could pollinate, every disease they could help abate, that entire ripple has been taken out of the wild.
you just sliding doors
Yes.
that whole bird's life, that
Correct.
whole like the lineage that bird might have.
Yep.
Yeah.
So that's rough.
Yeah. All right. From rescuing platypuses to don't feed the animals, let's talk about black hole sun. Well, it's a black hole star. Maybe. This is a really cool story. And I remember when the James Webb Space Telescope first started showing images, people were talking about, astrophysicists were talking about how there were these red dots in the background, like far away, way back in history. There were these red dots in the images, and they didn't know what the red dots were. And now researchers think they might know what the red dots are. Of course, it's still a hypothesis. They have done computer simulations and analyzed things every which way. So we haven't gone and visited one, so we have no way of knowing. So what they think these red dots in the James Webb Space Telescope's images from the early universe is a new type of astrophysical object. It looks like an enormous red star, but it has too much energy. Energy similar to that of a black hole, about 100,000 our sun masses. And so these researchers put this there. Well, maybe the red is dust from like a big star area. It's so far back in the history. Is that dust? What is it? Could it be hydrogen? Anyway, they realized at way more energy, these red dots are blasting out way more energy than a normal star would. And they are big. They're like the size of a solar system. So they're really big in their area. These red dots, their analyses, they believe that there is a central black hole surrounded by a cocoon of hydrogen gas. And the hydrogen gas is part of it's getting heated, and it's also kind of blocking all of the energy that would initially be emanating from a black hole if it just didn't have anything around it. And so they are really putting forward this idea that it might be a black hole star. And that's what they're calling them now. So the red dots, they are saying they're calling them black hole stars. They looked at the signatures, the energetic signatures of the red dots. The dots were red, they were bright. They were like, okay, so the red light, that means a few things. So it could be a veil of dust, right? They also were looking up at the pattern of the dust that they were seeing. But it was like there was a complete cliff when the wavelength of the light that's being detected was considered. And so below certain wavelengths, it just completely goes off. And this is a spectral drop off known as a Balmer break. And so this, they know, is usually associated with dense gases in the atmospheres of stars. And so they were like, this is a really, really big break. And so then they were like, is it a different stellar atmosphere on a bigger scale? What's going on? And they really think it's a dense screen of hydrogen gas. And that this dense screen of hydrogen gas is what, it's early universe, so that makes a lot of sense. It looks like a star, but it's super bright, so it can't be a star. How do you get that? Not nuclear fusion. It's something bigger, a black hole energy source. And so because of the cocoon and the way the light is coming out and the way that we're seeing it, and the fact that it looks like a star, that's why they're calling it a star. It's kind of their shorthand, but it's not really a star the way that we think of them. It's a black hole surrounded by hydrogen gas. And they think this is what explains these red dots that we have been seeing. So they have this one object they've been looking at, MOM BH asterisk -1. And this is black hole star 1, and this is the first of many of them. And they have been looking at a bunch of these little red dots from the JWIST images and think this is really the thing that explains it all. Their title of their study is a gas enshrouded, I love all this, gas enshrouded and gas reddened black hole at cosmic dawn. I mean, that's not sensational at all, but these some of these images from their spectroscopy are really quite wonderful. So I'm going to share these images right now with those of you who are watching on the video stream. We've got these really great energetic visualizations of the red dot in one of this black hole star number 1, and the energy cliff that we're talking about taking place at the energetic absorbance of hydrogen. So they've got some really great spectrum detail from NIRCam and MIRI, and they're really looking at these things. I think the images that they've got here are really, really beautiful. Yeah, I know it's quite a headline, right? But I haven't seen enough people talking about the use of the use of, instead of black hole sun, black hole star. I mean, they're so close. They might have copyright issues when it comes to songwriting, though. Anyway, they've got this explanation. And of course, it's going to be tested and tested and tested. But this is what publication is for. They want to put all their data out there so that other people can tear it apart, come up with other ideas, and we can see, how close this is to reality. But this is the first really good explanation for these red dots that we didn't know what they were once upon a time when JWIST first started taking its pictures. Could these objects be the seeds of galaxies? I think that's a really interesting question. I think that if there's no dust involved that could be ignited, I think this is where, because the seeds of galaxies, you want to have more than just the gas coming together, you want the dust as well. But maybe the energy, this is a great question, Paul, maybe the energy from the black hole can energize the hydrogen atoms to interact with each other in a way that would lead to chemical changes that would impact element formation. Because there is a big question that we've been having about our theory of element formation. It's the whole idea of the whole question of is it just the explosions of supernovas that lead to the creation of heavier and heavier elements? And so maybe this has something to do. I'm speculating. I have no clue. Yeah, and Kevin Reardon is saying these are at the furthest resolution of Webb. So exactly, we can't go in any closer and figure out what they are. This is completely hypothetical. They've done because of the light spectra and the instruments on JWIST, we're able to get some really good information about absorbance spectra. And and emission spectra and what we're seeing there. But in terms of really good high resolution imagery, definitely not there. But I don't know. I think it is oh, and one by land, two by sea is saying the fifth explanation by far. But yeah, I don't know. I think it's the best that I've heard so far. May the 5 hypotheses challenge one another. This is the best part of science. Coming up, especially the theoretical astrophysics, coming up with new ideas. Yeah, the black hole is not going to blow apart. Absolutely not. It should just, according to the way that black holes work, it should just be a black hole slowly emitting its energy for a long time. But it's emitting all that energy that is energizing the space around it. And so it won't blow apart. No, but maybe the energy, because it's such an intense amount of energy, maybe that that energy intensity will change things. This is something we have no idea. This is yeah, it's a great question. And Dallas Middleton is asking, why do they only show up in the furthest part of where we can see, and why wouldn't they exist later in the timeline? And that is a great question. Perhaps because there's no reason for there to be just kind of lonely hydrogen gas as often around black holes in our more complex, clumpy universe that we have now versus the more dispersed universe that was picking up speed back at that period of time. I am talking out of my butt. So I have no idea. And I think these are wonderful questions. Maybe we need to get one of our favorite physicists on to talk with us about this and to give us a little bit more information. Yeah, it'd be wonderful to see where if this idea is correct, where it fits in the timeline. Okay, moving on from black hole stars, I was hoping Blair would be around to talk about this story, but she's taken a very quick break. And so I will move forward here into our story about bacteria and female reproduction. Apparently, the researchers just publishing this last week in Cell Host and Microbe have shown that lactobacillus bacteria species that exist in the endometrial lining of women declines with advancing reproductive age. And they were able to show that as this process moves forward, the aging endometrium has, more inflammation, it has oxidative stress, it doesn't receive any embryos, fertilized eggs, etc., into itself easily. And so this is the problem. As women age, it becomes harder for little zygotes to embed into the aged endometrial lining. So they were looking at this study and were able to see that there are multiple lactobacillus strains that exist in the endometrial lining. One in particular, L. gasseri, becomes less active as women age. It is the one also that is tied to inhibiting pathogens, anti-inflammatory activity, antioxidant activity, and also adhesion to endometrial epithelial cells, which is very important for, for the grabbing onto things, making sure that the endometrial lining can grow thick and productive. Anyway, they did some work to look at the mechanistic action of L. gasseri and how it works in the aged endometrium. They were able to show that when they increased it, it changed a certain signaling molecule and the way that it signals. And this is really what they call it, hipposignaling. So in the aged, untreated state, hipposignaling overactivation leads to decreased YAP going into the nucleus. I'm not making this up. After L. gasseri is added, they found that led to normalized hippo normalization, normalized hippo signaling that increased YAP uptake into the nucleus. And YAP apparently is very important for these processes that are involved in the endometrial lining being able to accept a fertilized egg, a zygote. So anyway, they tested it in mice. And what happened? Dun, 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 when they gave bacteria to aged mice, they showed that they had increased implantation rate. So increased number of implantations. And that could lead to more offspring. So one way that as people are getting older, women are getting older who maybe you're doing IVF, maybe you're thinking of IVF, maybe you are, taking hormones, maybe you're trying to figure out how to get everything to work. There's this new line of research that suggests that bacteria might be part of creating a better system within your body for supporting pregnancy. So anyway, I think that's so exciting and interesting because why do we ever think of bacteria as being part of something like pregnancy? We talk about the gut-brain connection all the time, never talk about it in terms of how it might support or inhibit or detract from the process of pregnancy. Doop, doop, doop. Okay, moving on to my last story for the first part of the show. This story I think is so great because it has a Tolkien theme. Researchers discovered in Pacific Ocean seamounts. They have discovered a whole new species, not just a whole new species, but a new family of corals. So I just feel like this is incredibly wonderful news because number one, we talk on this show all the time about climate change and how corals are dying out. And we keep talking about the issues, the problems related to coral reefs and their survival. And, and they're beautiful places on this planet. The biodiversity and the life that coral reefs support it's just incredible. And it is essential for healthy oceans. And to have discovered not just a new species, but a completely new family of coral, I think is a very, very exciting advancement. So this is a new octocoral family. So octocorals, Anthozoa, Octocoralia, Malacalcionacea. These are this new octocoral family was discovered from seamounts, like I said, in the tropical Eastern Pacific. Now, these are places where there's volcanic activity, where these mounts come up. There's lots of diverse biodiversity around seamounts very often. And in this particular case, there were a couple of oceanographic campaigns that went down off the insular shelf, is what they say, of Isla del Coco in the Pacific margin of Costa Rica. And they identified a bunch of new taxa, which is really cool. And so this particular study comes from work based out of this 2019 trip and another in 2023 to confirm the findings and collect samples. And they were able to show that the aspects of this coral are unique. They do not fit within the phylogenetic definitions of current extant coral species. And so this is just a very exciting study because they did multiple phylogenetic analyses within the order Malacalcionacea, and they were able to keep working on through getting genomic data, multiplexed reads, doing the genomic good stuff to parse and select and figure out where everything needed to go. And they have determined that now this is under order class Octocoralia, order Malacalcionacea, family Laurencaidae, genus Laurencae, and it is the species Laurencae elenia. Now, I love this because the picture of the octocoral that we have is this beautiful, fluffy, kind of leafy, golden coral. And it's sitting on what looks to be a reddish rock, but what is actually a bunch of teeny tiny bristle stars. These are little brittle stars, and they are all over the place. A lot of them covering the rock or whatever is coming out of the seamount, and the golden feathery appendages of Laurencae are emerging from the bristle stars. And so the researchers named, using the Elvish language from Lord of the Rings, and this is another reason why I would love Blair to be here right now, because she had a Tolkien story coming up as well. So much Tolkien in the show right now. Laurencae, it means golden tree in Quenya or Kenya, the Elvish language. And so it is referring to, of course, the bright yellow tree-like colonies, these feathery tree-like colonies that are growing. And they say within this paper that was published, the golden octocoral gardens on fields of brittle stars evoke the mystic world of elves where they spoke Kenya, their ancient tongue. So anyway, yep. Elvish coral, everyone. Tolkien from the deeps of the Pacific Ocean seamounts. And we have beauty still being discovered, new species and new families still being identified in the world around us, even as we could talk about so many other things going missing. I just like looking at it. The field of bristle stars. Anyway. This is This Week in Science. Thank you so much for joining us for another episode of this science program that is all human produced, not AI. We're doing it live. We're talking about the science. We're curious about how the world works. We're so glad that you're curious too. If you are a curious person and just want to find out new things about how things work and have new questions to ponder late at night, early in the morning, I don't know, while you're brushing your teeth, eating your lunch, whenever, I hope you continue to enjoy This Week in Science. And if you are able, please consider supporting us on Patreon or purchasing something through our Zazzle store. We are listener supported, and so these channels are the ways that we're able to really keep this going. If you are able to do that, please head over to TWIS.org, click on the Patreon link or the Zazzle store. The Zazzle stuff makes a great gift to some other wonderful TWIS lover in your life. So maybe here's the process. Find a friend this week, get them hooked on TWIS, and then you can go to our website and buy them a TWIS gift. And you've done double duty for the show and for your friend and for yourself, because you've also expanded our community of amazing people. And I really have to say that this TWIS community through the years has been wonderful. It's a real joy to be here every Wednesday talking to you all. So thank you for being here. Thank you for your support of This Week in Science. We really can't do it without you. Okay, we're going to move right into monkey brains. Well, not monkey brains, because that is inaccurate, as I know anybody who is a listener of this show will remind me. Primate brains. The news from this last week, after our last week's show, when I talked about dodos and their brains and the use of fossil skulls from dodos to be able to look into how their brains maybe worked. So what senses they relied on more to infer dodo behavior. Well, Duke University researchers led a study that was published this last week in Science on using fossil evidence, so skulls of multiple primates, to support the idea that in the primate brain, we've always been like, ooh, the frontal cortex, it got so big, or the neocortex, right? The part of the brain responsible, or in primate evolution, that is initially responsible for our cognitive prowess on the planet. Well, I'm just going to be braggy there a little bit. Their study suggests it has nothing to do with the frontal lobes of the brain, but more specifically to do with vision, and that it is because of vision and the need to see clearly, maybe to visualize, maybe to find food and hunt, maybe to have imaginings of visualizations, maybe to understand things, maybe to have, broad abilities when it comes to vision. Our visual cortex is making up for a very dumb eyeball. Our eyes are dumb little fluid-filled sacs in our heads. They do work, of course, but our visual cortex is everything about visual processing. We would see a completely different universe around us every day if it weren't for our visual cortices. And so what these researchers have done is they've like, well, vision is really important. Visual cortex in primates is pretty big. So let's see how it evolved, how it changed over time in different species of primates compared to the frontal lobe and to other areas of the brain, and with respect to brain size in general. So is it that everything's expanding at the same rate? We already know that's not exactly true. So they were looking at all these different factors of the changes in brain size over time. Now, they were able to use not just the Duke Lemur Center Museum of Natural History's collection of skulls, and then they were also able to use Duke Shared Materials Instrumentation Facility to do high resolution micro CT scans and reconstruct 3D models. So they had a number of digital models after they scanned all of these skulls. So to be able to look at these endocasts over time and also use digital tools to be able to look at the scaling, right? So, okay, we know how big the inside of the skull case is. We know how big that imprint of the frontal lobe is for a tarsier, for example, or the visual cortex for that animal versus a lemur. The researchers say everything from humans down to tree shrews, they all fall in the same line relative to the size of the brain. The proportion of the frontal lobe is a constant. And when they looked at the occipital, parietal, and temporal regions, which are parts of the brain that are specifically or highly used in processing visual information, those expanded rapidly. So tarsiers, anthropoids, this is monkeys, apes, and humans are the anthropoids. As we went out along the branch of the primate family tree, as the optic nerve grew larger, so did those areas of the brain. And so basically, it's a quantitative study of these ancient fossils of a whole bunch of primates by taking advantage of this really interesting collection of old skulls and modern technology. It's really been able to look at the small changes. So the fact that you're looking at the size of the optic nerve, because they're able to see the imprint of the optic nerve in the bottom of the skull case. I mean, this kind of stuff is really impressive and amazing. But the fact that this is, the first time that we're really getting this deep of an analysis of this idea to be able to support the evolution of the primate brain as being led by vision as opposed to frontal lobe and attention, right? So it's like the different lobes are responsible for different things. And these researchers really suggest that visual sensory inputs are functionally tied to modular evolution of the primate neocortex. And if we think about it as well, there are so many areas where we have talked about we've talked about the brain and vision and language and how so many things tie together. And that's one of the big examples that they bring up, that vision is highly tied into cognition. So it's not just vision on its own, it's not just cognition on its own, or the neocortex on its own. It's that the neocortex could get bigger because the visual system was getting bigger. The visual system needed more space, maybe in the neocortex, for processing all of the visual information and actually applying it cognitively. So I think there are lots of ideas here that are fair game. But yeah, monkey brains. They took lots of pretty pictures, and I hope you go search for this study and the conversation that will no doubt come out of it, although it was a broad collaborative study between many research labs at a number of institutions, which really is the important way to do research these days. We get things done by working together and sharing resources, right? Okay. As we continue to move on in my stories for the evening, I hope you trust me. Trust is very important. And I remember years back, oxytocin became this magic hormone for making people like each other and trust each other. And suddenly everybody's like, I need to inhale oxytocin before I go to parties. I don't drink anymore. I just do intranasal oxytocin. It was a very weird time in at least American culture. I don't know how it went in the rest of the world. But researchers published this evidence that oxytocin can increase trust behavior in people. But that was like back in 2005. And since that time, there's been a lot of conflicting information. A study came out like several years later. It was probably like 2015 or so, where researchers did a registered study. And this is, I'm going to say, props for this group for doing registered studies, because this basically means they go, this is our hypothesis. This is what we're going to look at. They put everything out in the open in terms of their hypothesis, their null hypothesis, their alternate hypotheses, the stats they're going to use, how they're going to do the whole study before they even start. And so, I mean, it's done very well. Okay, so they did one study and they were like, it doesn't seem like that original data bears out. Like, we've seen stuff in other studies that doesn't seem to work. And our registered study basically says there's not a general effect. There's an effect, but we don't know exactly what it is. It looks like guys who don't trust generally, who have a low trust profile, and I guess apparently that's a way that you are being judged, that people who have a low trust profile, that they're the ones who respond to oxytocin, that oxytocin increases in trust in individuals who don't trust very easily. So they said, okay, we've got our registered study, and this is an area we need to go and look at. So what did they do? Another registered study. And now we have the results. They have this new study. It's an international team. Ernst Fehr from the University of Zurich, Bodo Vogt and Paul Bengart, Otto von Guericke University in Magdeburg, Caroline de Klerk, University of Antwerp. They registered this study ahead of time that involved about 360 men who were rated as having low trust profiles. So they basically went out and asked people questions, and they're like, ooh, you don't trust people, be in our study. Anyway, they only did this study on men, so it can be compared to earlier oxytocin studies. And obviously, other studies moving forward will be either have female women or other ratios of control groups, but their control group this time was basically oxytocin or a placebo, no oxytocin. And then they participated in a trust game. So under anonymity, it was like, okay, you're anonymous. This other person's anonymous. How much money do you want to give them? Asked him first and then gave him oxytocin and then asked him again, how much money do you want to give to this anonymous person on the other side of this computer system? They were able to show that compared to their earlier study and pooling the data together, they were able to show that when low disposition trust men had oxytocin doses, their trust increased about 17%. So that's, and they're saying trust is how much money they would give people. So basically, they would give people more money, 17% more money when they after they'd had oxytocin. So what they have shown pretty well is that oxytocin has a selective effect. It does increase trust in people, but not all people. And it's not just you take oxytocin and become more trustful. It relies on context, whether or not the disposition of an individual to trust is low or high or whatever. And so what they've really been able to show also is that within that low trust disposition, it doesn't matter where you are in the low trust disposition, slightly low or highly distrustful. If you are in there, you're still going to be impacted by oxytocin. But the questions are, how does it work? What exactly does it do to the brain? And the researcher Fair says it's possible that the hormone intensifies the expectation that others will cooperate. And so this kind of goes back with like another study that I was talking about a couple of weeks ago that was related to oxytocin and pretty much related to, how people are going to anticipate results come out. Blair!
Hi.
Are you back?
I'm back.
Yay! Oh, what an
Did I miss everything?
Not everything. I have not told the one story of the LLM study we didn't know that we need, but we really did. We really did. But I can save that for after Blair's Animal Corner if you would like to move on. I've been doing my thing. I will save my one last story for the cherry on top at the end of the show.
Perfect.
Yes, but now it is time for that wonderful part of the show that we love to call Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh my goodness. Well, let's start with the frogs. I have some analysis paralysis.
Analysis paralysis of whether or not you should talk about the frogs?
No, the frogs have analysis paralysis.
The frogs do. Oh my.
Yes. So frogs, they call for each other when it is time to mate. Female tree frogs, they like to pick their most desirable mates. And so this is looking at Cope's gray tree frogs. They gather on ponds during mating season. They listen to large groups of males chirping and noisy choruses, and then they pick the most desirable male. What they found is that if the pond is too crowded, then the females will get choice overload, and they will struggle to pick the most desirable male. They actually think that this choice overload is influential in the evolution of the species over time. Because if they always picked the longest, most desirable call, then the shorter calls would become less and less prevalent. But because of the choice overload, they posit that this allows the females to continually make the quote-unquote wrong choice and allow those genes to perpetuate into future generations.
Oh my gosh. So, oops, I made a wrong choice.
Well, it's just like, it's so hard to pick. Oh no, I got pistachio ice cream.
But
could have gotten vanilla, could have gotten chocolate. Got the pistachio.
But that's not an issue that will likely impact your future chance of success. Aside from,
so
if you're
really,
allergic to nuts.
That's the question. So, the question is, if they are picking the best mate via the best call, is there a relationship between the longest call and better genes? Now, you would assume there is, because that is what the desirable call is, right? Like, otherwise, why would that be the desirable call? But unclear as to what kind of link there is between the longer calls and whatever genes are quote unquote better. The test study is actually really interesting. So they had female frogs in an enclosure. They had a series of speakers spread around it. They played recordings of real male frogs calling. Females chose their preferred call by hopping towards the speaker. In previous research, they showed that they do prefer the longer calls. The team played these mostly longer target calls, and then they would have shorter distraction calls. They observed that the more male calls there were, the more confusion there was from the females. When they had just a couple options to pick from, they pretty much always picked the longer call. But when they had to listen to 8 different males at once, they selected the target call only 25% of the time.
Which is way less than chance.
And in some cases, the females got so overwhelmed that they didn't make a choice at all. And girl, same.
Nope, too much. This is why I don't go places like IKEA or the big box stores. Too many choices.
Too much. That's why online shopping is so overwhelming. And I do really miss just going to a store, having 3 options and buying a thing. And now instead I spend 4 hours on the internet and buy nothing.
I'm so glad I'm not alone.
Yeah. So to make sure that they weren't just having trouble hearing the desirable call, they actually ran an identical experiment, but with background noise. So there were male calls overlapping, but then they were in a noisy environment. So that kind of replicated when there were 8 different calls. So similar kind of like decibels. The noisy environment didn't have an effect on the females' choices. So they were instead struggling with choice, it would appear. Choice overload shapes the genetics. Evolutionary theory, for example, suggests that if females have strong preferences for males with long calls, the males with short chirps should disappear, but they haven't. And so this is the explanation. Choice overload could potentially keep males with short calls in the gene pool by causing enough confusion that females choose a mate who is in theory less optimal.
So did they explain at all how many different calls do they normally hear in the wild?
Question.
Right?
Good question. Yeah.
Is it 8, 10, 20?
Well,
like
not to sound like a broken record, but like with habitat destruction and climate change, you could end up with more frogs in one area because the pond is smaller, because there are less ponds, and so you could end up with kind of more overload than there has been in the past. Potentially, I don't know.
So hypothetically, speculatively, it could lead to continued diversity within the male frog population,
Mhm.
and the songs that are sung, but it would not necessarily be great for overall population longevity and growth.
Well, so I mean, but here's the thing, like, why are there sneaker jackfish that like can get by the big impressive male and can mate with females? Why are there sneaker males in male dominant society, or mating
Yeah.
groups? So like
Alpha male dominant, yeah.
yeah, so like why evolutionarily would you need that? And my argument would be genetic variation. You just generally need genetic variation. So if there is a benefit of being like, hey, if I call really loud next to this really impressive male, maybe I'll get a mate instead. Like, that is also an evolutionary advantageous method. And so why does that continue? It's because their genes that they are passing along can't be that deleterious, or like those babies wouldn't survive. So their genes they're providing seem fine. I guess.
It's okay. Hey.
But, variation makes the world go round, as one would say. But anyway, I just thought it was very funny that female frogs have analysis paralysis.
But so going back to that with the analogy of a crowded pond ecosystem, right? If there gets to be a cacophony that's too loud, aside from just diversity, the other extreme option is that females don't mate.
Yeah, and I don't know if that would be at all. Like, it would probably be like, not tonight. Yeah, headache, or it could be that they wait around until some of the males leave, and then they make a choice. This was in a lab, so it just kind of had a very clear start and stop time. But I think, outside of a vacuum,
they need to put the computers in the ponds.
Yes.
That's the only way we'll know.
I am highly in favor of LLMs working on synthesizing data from field cameras and sonic data and all that good stuff. Like, that is exactly what these things are for. That's what the machine learning should be for, is for synthesizing data.
It's way faster than the many years of graduate students doing it over and over again.
And all the citizen scientists that are kind of sort of trained, but not really.
But not really, yeah.
Yeah, do you want to talk about snail slime?
Yes.
So,
but not just one snail slime per species.
I foolishly assumed,
you are not a fool.
Snail slime was just snail slime. Did you know they can make different slimes?
Of course I did not.
Snails do not produce just one type of slime. They can vary it as needed. They can make it liquid, solid, sticky, slippery. How? How does a snail produce completely different material properties using the same building blocks? This is a study looking at snail mucus.
We all love mucus. Yeah. Mucus is such a wonderful compound.
Anyway. It's important. It keeps us from getting sick. It keeps our turbinates wet. Lots of good stuff. Anyway, researchers analyzed 5 different types of snail mucus. Mucus that enables locomotion, mucus that acts as a strong adhesive, mucus that protects the animal from dehydration, mucus that seals the shell during prolonged periods of rest, and mucus that serves as a defense mechanism.
I think I've seen that the hardening of the mucus.
Yes. So I'm going to do that again. So we have
say it again.
Locomotion, adhesive, protecting from dehydration, a shell during periods of rest, and a defense mechanism. These are not all the same mucuses. Researchers found that the snail consistently uses the same components for the different types of mucus. mixing them in varying proportions. The chemical building blocks are proteins, mostly collagen and calcium.
Collagen.
Yes.
Okay.
They found collagen 6 as the key component of mucus. This is known for its structural role in human skin, bones, and joints. Together with disordered calcium carbonate, which the snail stores in its glandular tissue and releases along with the mucus as needed, they adjust the proportion of collagen 6 with the calcium carbonate, and that plays a decisive role in controlling the properties of the mucus. The snail uses the total amount of proteins and the proportion of collagen 6 to alter the density of the protein network in the mucus, which has a direct effect on its mechanical properties. The denser it is, the tougher the mucus is. Calcium serves as either a cross-link in the mucus or as calcite to reinforce the material. So that's that like harder shell that's high in calcium carbonate.
So I'm just looking right now at, of course, I'm, as you're talking, surfing the web looking for information on snail mucus.
Mm-hmm. Yes.
There was 2019 work on the use of snail mucus or snail slime for medicinal uses.
Mm-hmm.
And so, not just eating snails, but using snail slime for skin inflammation and treating wounds.
It's a common ingredient in face masks.
Face masks, right?
Yeah.
So, but the question is, now that you're talking about the differential purposes of slime, do the people using the snail slime for face masks know which slime is better for your face mask?
Yeah, they need to
is there
prod the snail just right to do the right kind of slime.
It's not the sexy slime, it's the defense slime.
Yes.
Or, you need to have a strong trail slime. I don't know.
Mm-hmm. So, of course, that's one of the things that the researchers said at the end of this was that, understanding how these natural materials are made has a variety of potential functions with very few building blocks. Understanding those principles has a great importance for developing sustainable materials. Potentially, they could produce environmentally friendly adhesives, functional coatings, materials for medical applications like you're talking about, right? But I think for me, the thing that I'd really want to know about next is what the trigger is to change the makeup of the slime. Like, is it
what,
like
what vibrational
stimuli? Is it like, is it something they're sensing through smell? Is it like, does it have to do with how moist the air is? Like, what is happening that is triggering the snail to change their mucus?
And how fast can it shift? So how fast is the shift in that mucus production, right? Mm-hmm. I'm going from traveling down a tree branch to I found another sexy hermaphrodite, and let's have snail time. So
yeah, I want to dangle.
Like the slugs, yeah. But how fast can that happen? I mean, obviously, snails do not move at the speed of sound, so we're not looking at fast changes being necessary. But how relevant to the speed of a snail are they?
Yeah. No, that's a great question. I mean, and can they switch back and forth?
Yeah.
I love this idea that like every time I see a slug or a snail now, I'm going to be like, what kind of mucus you making?
What you doing? How much calcium's in there?
Got?
Yeah. Is that strong?
But that's also like, it reminds me too that like, if I was around people who would like poke a snail or a slug, and then they would start, it looked like they were oozing, and you'd be like, you killed it. No. It's like, well, maybe they were just releasing defensive slime and they went off into the bush and they were okay.
Yes. It's really, really only bad if they stepped on a snail and that was the ooze.
Yes, but
this is cool.
Yeah, I just went on a hike with my family a few weeks ago, and we probably saw 100 banana slugs. And now I feel like I have to go back just to be like, What kind of slime you got?
And so, since snails are doing this, is that this? Did they look at slugs as well, or was
No,
it
they just looked
only
at this
snails?
one species,
One species.
common
Okay.
brown lip snails. I think it is fair to assume
Yeah, I would assume,
all
but
snails
they
and
have,
slugs.
but they haven't done that work yet.
I don't know if it's 5. I don't know if the makeup would be exactly the same, but I think it is very fair to assume that all snails and slugs create variable mucus.
I'd love to know like the difference and how it shifts based on slug snail social structure.
Yes.
Like, how does that work?
Well, because yeah, like
Oh,
the
cool.
dangly sexy mucus is definitely a 6th that we didn't discuss in these different types, which like what is the makeup of that? Because you're carrying the entire weight of the slug.
Yeah. And you're building those threads together with another slug.
Yes.
And yeah, it's a
yeah, and if anybody
definitely
listening
not a
doesn't
spider
remember
web.
this, go do some Googling later and look at slugs that I can't even get
Copulate
fully
while
into
dangling.
It. Yeah, they copulate while dangling. They do it via like basically a genital sword. They like stab each other in the head and inject sperm directly into the body of the other slug. It's fascinating. But yeah, look into it. It's a very interesting wormhole, slug hole that you could fall
Totally.
down. I've spent many an evening in that particular corner of the internet and literature. Anyway, yeah, so don't act surprised, Kiki. You know who's on the show.
Blair with her evenings down the slug hole.
I have a book that is literally all about invertebrate sex.
That's cool.
So
it's so good.
That's the whole deal. Anyway,
it's a good deal.
Regardless, next time you see a slug or a snail, say, "Hey, what you secreting, buddy?"
I will take you up on that. All right, my last story for the night.
Yes.
What I wanted to talk about this publication out of iScience, researchers using LLMs, large language models, to generate personality questionnaires, personality quizzes, and also analyze those personality questionnaires. The researchers in their work, and the reason that I'm going to get to why I think this is the study we didn't know we needed, and it is a reason that is not highlighted as strongly in the paper as I think it should be. The researchers used what they call the 5-factor model, the Big 5, and this is a hypothesis that personality traits are encoded in language. They used LLMs trained on the DSM-5 for personality disorders section and a popular astrology book to generate questionnaires, administer them, and also analyze the results. What they found is that the LLMs trained on the DSM-5 and an astrology textbook were able to use the scientifically proven or evidenced DSM-5 very well to support personality types and to create questions that stemmed from the various personality disorders, and to be able to actually analyze the results based on the various answers that would be given. So if a person gave one certain result, it'd be like, oh, hey, that suggests paranoia or not paranoia, blah, 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 the other way. When they did this with the astrology textbook, they were unable to show any correlation to the 12 astrological signs that have been purported to be true by astrology for a very long time. And so the astrology and in the book, it's in the paper, it's wonderful. The researchers do say in the introduction, astrology is included as a contrast case designed to probe the role of source structure, independent of scientific grounding.
Nasty.
Yeah. And so in the conclusion of their paper, what they were able to show is that, yeah, these LLMs can use scientifically evidenced personality measures that are based on psychological scientific studies, and to be able to create consistent and useful personality questionnaires. And so this kind of they used validation measures to show that the questionnaires were actually, they were working to show what they wanted to show. What they also were able to show, though, is that the models, the LLMs, based on the results, could pretty much give you a result of what they think you're going to answer before you answer it now. So watch out for that one. But in relation to the astrology situation, they did find that there were certain aspects of, astrological information that, similar to DSM-5 information, can be generally applicable to proportions of the population. But there was absolutely zero consistency related to you're a Leo, a fire sign, a star, your star sign, and anything that came out of this. And so really, if you're going to pick any LLM study that you want to use, like for evidence of usefulness of LLMs, I think it's this one.
Yeah, it's pretty good. I mean, we know certain things like that. The arrangement of the stars and planets now during December is completely different from how it was when you were born. So, like, what is it actually?
And different from when the original star signs were created and started being used. It's so many years.
So, like, what is it even? Anyway. And it's all based on the Gregorian calendar, which is all kind of make-it-y up anyway.
Yes.
And so it's
Yeah, so this study did not, its effort was not to show anything evidentiary about astrology, really, right. But I think they did, and they did not highlight it in the way that they should have.
Yeah. No, I think Paul Disney's right. Like, it was the world's meanest control. So, like, basically they used it as like, we all know this is BS, so that's our control.
That's our controls.
Yeah. But for real, if people don't believe in real, like, humans, maybe they'll believe in LLM if they tell them that homeopathy is mostly bunk and, like all these other things
that's the scary part of where we're going now, actually. Yeah.
Yeah. If they ask their ChatGPT do vaccines cause autism? And ChatGPT says no. Will they actually vaccinate their children?
You know what the weird thing is? They're probably more likely to trust their ChatGPT.
Yes.
As opposed to, say, now the actual CDC or their own doctor.
And it's because it's a machine, there's this facade of impartiality.
Yes. Yeah.
But it's actually super partial because it wants to tell you exactly what you want to hear.
What you want to hear.
It's sycophantic. So it's like, yes, exactly. You're so right.
My favorite trend on the social media video thingies now are the people who are using the different chatbots to talk to each other.
Oh, sure, yes.
One I saw recently, a young man was going to meditate and was ringed by iPhones, all with whatever chatter on there, and he said, "I think I would like to meditate now. Can you help me?" All like 12 of these devices pinged it. Yes, well, of course, meditation. And then they were just talking to each other. And it was like, there's no meditating here right now.
Yeah. Well, that's even like when you ask them to count and they'll just go like 1, 2, 3, and we'll just keep going on like that forever. Right. It's like, no, keep going.
You don't have to talk about how you get to the next number. Let's just keep counting. Just go to the next number. It's okay. Oh man, yes. One by land. CDC is an LLM now. I think that's a t-shirt. Oh dear. Well, that does it for me for stories. If you had anything else you wanted to talk about.
Nope.
Anyway, question the personality tests you're taking in the future. You don't know who's giving them, but
I know. Well, it's like, do you remember all those silly, always BuzzFeed quizzes? Those were like, what kind of unicorn are you? Right?
Those were the beginnings of training these models.
Yeah. And unfortunately, like if you try to do anything like that now, you're going to be feeding your data into an LLM that can sell your information to.
Yeah, it wasn't LLMs back then, but it was still it was the beginning of data models. And so any quiz that's like, what's your middle name? Just run. Stop answering any quizzes online.
What month were you born?
You guys, correlation, large language models. There's so much information. Just stop. Do not talk to strange quizzes, okay?
Do not talk to strange quizzes. Do talk to strange snails, though. I would recommend
talk to the snails. Yes. Go outside. You can touch grass and talk to snails.
Yeah.
Yeah, it's good. All right. I think we've made it to the end of our show. Have we made it?
Yes.
All right. Everyone, thank you so much for joining us for another episode of This Week in Science. Everyone in the chat room, you're amazing. I love seeing your chat. It's so good. So many comments that are pinging into my brain as we go through our conversations and how we talk about everything. Thank you for being here. Thank you for being a part of the show. And thank you to certain people for really helping to keep the show going. The chatters are a big part of it, but we've also got people like Fada, who every week helps with social media and our show notes and is absolutely amazing. So thank you, Fada, for all that you do for the show. Additionally, thank you to Paul Disney for recording the show. Thank you to Arnlor Gord and other people who really help to keep our various chat rooms great places to be. I mean, it's by being good people that we maintain good places to be. Thank you for all that you do. Rachel, thank you for editing the show. And thank you so much to our Patreon sponsors. Thank you to Aaron Anathema, Adam Mishkan, Alan Viola, Ali Coffin, Andrew Swanson, Artem, Arthur Kepler, Bob Calder, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Derek Schmidt, Don Mundus, E.O., Eden Mundell, Eric Knapp, Flying Out, Fredis 104, G. Burton Lattimore, Greg Briggs, Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Kenton Northope, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenflow, Mark Jenkins, Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Pierre Velazarb, Richard Badge, Robert Norland, Robert W. Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Clarence Lamb, Shubru, Stephanie Embry, Stephen Albaran, Steve Leisman, a.k.a. Zima, Sue Doster, Teresa Smith, Tony Steele, Vegard Shefstad, and What the If? Thank you all for your support on Patreon. Patreon is pretty much how we pay the bills. So if you are able and interested in helping to support the show, head over to TWIS.org and click on that Patreon link. On next week's show,
we will be back Wednesday at 8 p.m. Pacific time, broadcasting live from Twitch, YouTube, and Facebook.
Yeah, that is right. And if you want to listen to us as a podcast, just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe too. We like your friends.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. You can also sign up for a newsletter.
Yes. We love your feedback. If there's a topic that you want us to cover or address or a suggestion for an interview, let us know. One of our social media accounts at TWIS Science, TWIS, whatever, we're out there. And you can send us emails too. But if you do, just put TWIS in the subject line so your email doesn't get spam filtered into where, Blair?
Into a pile of snail slime.
Ew. I will not be digging it out.
We look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.